Cek, cek. Cek. Oke. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome to guest lecture Organic Natural Product Chemistry. Respectable, the lecture program in Chemistry Education FKIP Tanjung Pura University. Honorable guest speaker in this event, Mr. Professor Mario A.Tan, PhD. Honorable all the participants and of course all invited guests. First of all, let's say thank to Allah Subhanahu wa Ta'ala who has give us guidance, happiness, healthy, and mercy so we can attend and participate in this special event without any obstacles. Praise and salutation up on our Prophet Muhammad Sallallahu Alaihi Wasallam who had brought us to the path of light from the darkness in this life. Ladies and gentlemen, my name is Dea Aninda. It is an awesome and precious chance for me to be your master of ceremony in this morning in our special event, the guest lecture Organic Natural Product Chemistry, the chemistry of Pandanus Alkaloids. Ladies and gentlemen, on this special morning, we have several agenda as follows: opening by the MC, national anthem, pray, welcoming speech by the head of Chemistry Education Study Program of FKIP Tanjung Pura University, the lecture by Professor Mario A.Tan, PhD, discussion session, certificate and photo session, and closing. Well, ladies and gentlemen, let's begin this event by reciting Basmalah. Bismillahirrahmanirrahim. Move on to the next agenda is listening the national anthem. Attendees, please activate the video mode. Okay. Next is welcoming speech from Mr. Rahmat Rasmawan MPD as the head of chemistry education study program of FKIP Tanjung Pura University. To Mr. Rahmat Rasmawan MPD, time is yours.
Is Mr. Rahmat here or may I ask Mr. Tulus Junanto to represent our study program?
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you for Professor Mario Tan. Glad for us. You can give the new science for me, and this is good for our students to get the science from the other country that we can apply it to our students, and hope your science, your knowledge can give us the new inspiration for to more our future, and good for our student also to represent their knowledge for the students also. And thank you for all the students who came attend this Kuliah Tamu, especially from Professor Mario Tan. And then good luck for us, and said Bismillahirrahmanirrahim. We will from Mario Tan give us the knowledge. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thus, the speech delivered by Head of Chemistry Education Study Program of FKIP Tanjung Pura University or representing. Thanks to Mr. Tulus for speech. Ladies and gentlemen, now we proceed to the lecture agenda. This presentation will be delivered by Professor Mario A.Tan, PhD, and also this agenda will be guided by Mrs. Dr. Rini Muharini, MSc, as the moderator. To Mrs. Rini, time is yours.
Thank you very much, Dea Aninda, as a MC today. Good morning, Professor Mario Tan, and all participants of guest lecture, Organic Natural Product Chemistry. Thank you for attending our guest lecture today. First of all, let us thank to Almighty because of His blessing we can come here in guest lecture filter meeting. I am Rini Muharini, one of the lecturer here in Chemistry Education Study Program, Universitas Tanjung Pura. So please allow me to be a moderator for this meeting. Our Organic Natural Product Chemistry guest lecture today, we will have Professor Tan as a guest lecturer. So may I present Professor Mario A.Tan from University of Santo Tomas, Manila, Philippines. Kumusta, Professor Tan?
I'm good, thank you so much.
Yeah, it has been more than 10 years since we have not seen each other, hasn't it?
Yes, yeah, I think that was in 2013. Yeah, I remember
Yeah,
2013,
yeah,
November
yeah.
2013. Yes.
Yes, yes, yes. So yeah, so I would like to introduce to all participants here, Professor Mario A.Tan, PhD. He's currently working in Department of Chemistry, College of Science, University of Santo Tomas, Manila, Philippines. And not a while ago, I think, 2021, he was appointed as Assistant Research Director, Research Center for Natural and Applied Sciences in University of Santo Tomas. His research interest is in organic chemistry-oriented drug research from natural products, including phytochemistry on the Rubiaceae and Pandanus species, bioactive metabolites from microbial sources, synthesis of bioactive small molecules, new protective potential of natural and synthetic compounds, and plant extracts against Alzheimer's diseases. So I think he's the perfect person and so expertise, and we would like to hear his expertise and experience to share with us. So please, Professor Mario Tan. uh, he received his PhD in pharmaceutical sciences in Graduate School of Pharmaceutical Sciences, Chiba University, Japan. Uh, he took MSc and BSc in chemistry in University of Santo Tomas, Manila, Philippines. Uh, he has lots of publication. Oh my God, 47 publications in international journals. Yeah, and he got awards, research grants, scholarship, fellowship. Uh, what about the secrets, Professor Tan? Could you please share with us?
Oh, yeah, the secret. Well, I don't know. Uh, well, it's
So
necessary.
yeah,
The academe, yeah, it's really necessary for you to publish. Yeah.
all right, all right. So we will have a lecturer from Professor Tan for about 50 minutes. I think it is. Is it okay? And then followed by discussion session around 30 minutes. So to all participants, you are encouraged to ask question by raising hand. You can use raise hand icon at the bottom of the Zoom. Yeah. Or you can also write your question in chats. Yeah. I will read it later for you. So to all participants, please activate your camera during this guest lecture because it would be polite if we can see each other here. Yeah. Thank you so much for your collaboration. So without further ado, so please, Professor Tan, time is yours.
Thank you so much, Dr. Rini. So thank you so much for, the kind invitation to share our work in the genus Pandanus alkaloids. So can you see my slides now?
Yes, very clear.
Thank you. Now, so for my topic that Dr. Rini gave to me would be about the alkaloids, in particular, the genus Pandanus alkaloids. So just to give a bit of an introduction. Now, so when you talk about natural products, so these are naturally occurring small organic compounds. So basically, the molecular weight is about 150 to about less than 800 atomic mass units. And that these natural products are isolated from both the fauna and flora. So from plants, and then we also have isolation of the natural products from marine sources, from microorganisms such as the endophytic fungi, bacteria, etc. Now, these natural products are actually the source of the most complex and fascinating chemical structures, and they would represent natural biological activity, whether they exist as a single compound or a complex mixtures. Now, so why is the natural products, why is the importance of these natural products? Natural products chemistry is considered to be at the intersection of many fields, and they are an effective bridge from tradition to modern scientific developments, which would include natural products are being applied in ecological chemistry, in chemotaxonomy, combinatorial chemistry, chemical synthesis, in molecular biology and biotechnology, microbiology, biochem, and of course in pharmaceutical sciences through drug development. Now, there are a lot of pharmacological activities associated with these natural products, including, example, antioxidant, antimicrobial, anti-inflammatory, anti-cancer, neuroprotective, anti-diabetes, etc. So this slide would show you the outline of secondary plant metabolism. So as you know, we have different kinds of natural products. So you have the terpenes, you have the alkaloids. Here, you have the coumarins. Here, the one in the encircled, you have here the alkaloids, wherein the alkaloids are being biosynthesized using the amino acids. Usually, the alkaloids, they do come from the amino acids. Now, so based on that previous slide, so we have here the principal building blocks for secondary metabolism, which would include the shikimic acid precursor for many aromatic compounds, the amino acids precursor to alkaloids and the peptide antibiotics, and the acetate, which is the precursor to your acetylenes, prostaglandins, the polyphenols, and the isoprenoid terpenes, steroids, and the carotenoids. Now, by definition, alkaloids are highly reactive substances with biological activity. They have a wide range of pharmacological effects, including stimulant, constricting the blood vessel, or they may relax the smooth muscle cells. They usually contain nitrogen, wherein the nitrogen atom is derived from an amino acid. They have bitter taste, and in terms of characterization or isolation from various sources, they give a precipitate with heavy metal iodides. And the most common reagent used to detect the alkaloids would be the Mayer's reagent using the potassium mercuric iodide solution, wherein this alkaloid would give a cream-colored precipitate, or we use the Dragendorf's reagent, that is a solution of potassium bismuth iodide, giving an orange-colored precipitate. Now, in line with this topic, in line with the alkaloids, I'm going to present to you my research on the chemistry of the Pandanus alkaloids. And I am, as what Dr. Rini has said, I am connected with the Department of Chemistry and the Research Center for the Natural and Applied Sciences at the University of Santo Tomas in Manila, Philippines. So this is our university. So our university is like 21 hectares in total. It is located at the center of the capital of the Philippines, which is in Manila. It was founded in 1611, wherein making our university the oldest existing university in Asia and one of the oldest universities in the world. And we offer both undergraduate and graduate programs in various fields, so including humanities, arts, business and management, education, engineering, the health sciences, law, natural sciences, and the social sciences. Now, so I am, aside from being a faculty member here in my university, I am also a researcher, wherein my research interest is given in this slide. So I do work on phytochemistry, the total synthesis, and biological activity. So this is actually my research interest. In terms of phytochemical studies, I am interested in the endemic and indigenous Philippine plants. In particular, I am working on the different rubiaceae species, the Pandanus species, and I also work on the endophytic fungi. And then I do the isolation of these molecules, these bioactive molecules, And if something is quite important, we do total synthesis or synthesis. We do synthetic studies for structure elucidation of these new compounds, structure elucidation for the new compounds, in particular to elaborate its absolute stereochemistry. And also for in terms of the biological activity, for the biological activity, I am interested on the neuroprotective effect of compounds, pure compounds and plant extracts against Alzheimer's disease. But in terms of so Alzheimer's disease, as you know, would have a lot of biological hallmarks. So what I do study about Alzheimer's disease is that I focus mainly on the inhibition of the amyloid beta aggregation, wherein the amyloid beta aggregation is one of the pathological hallmarks or one of the most important or well-studied pathological hallmarks in Alzheimer's disease. So for today, I will be focusing on the alkaloids coming from the Pandanus species. So what are these Pandanus species? The Pandanus species are from the family Pandanaceae, wherein for the family Pandanaceae, it comprises of 4 genera. And the highest or the most number of species among these 4 genera of Pandanaceae would be the genus Pandanus. There are about 700 species of Pandanus, the genus Pandanus, distributed in tropical and subtropical regions. Out of these 700 species, 52 species are said to be endemic. And these Pandanus species, they exhibit a wide range of pharmacological activities, including of ethnomedical and pharmacological activities such as treatment against leprosy, rheumatism, headache as diuretic. They're being used for urinary tract infection. Sometimes they're also being used as an abortifacient, sedates mental patient, and as a root decoction against hypoglycemic activity, wherein that this particular activity is associated with 4-hydroxybenzoic acid. And of course, my interest would be the neuroprotective effect of this Pandanus species. So these are some examples of Pandanus species. You have here the most common here in the Philippines, and I think also the most common in Southeast Asia would be the Pandanus amaryllifolius and Pandanus nectorius. So Pandanus amaryllifolius, these are the Pandanus. This is the Pandanus species that we usually put in rice, or the Pandanus amaryllifolius is also the Pandanus species which is being used as food flavor. So we also have here Pandanus boninensis, Pandanus laevis. So Pandanus laevis is the very first Pandanus species which I work on during my undergraduate and masters. I'm sorry. Okay. And then Pandanus dubius is the Pandanus species which I work on during my PhD. And then of course we also have here Pandanus odoratissimus and Pandanus tectorius, which is one of the most common Pandanus species. So Pandanus tectorius is normally commonly found in the beach shores, you know, in the if you go in the seas or in the rivers, etc., you have a lot there of Pandanus tectorius. Now, in the field, it is very much easy to locate or to determine what is a Pandanus species, because all the Pandanus species are actually the same. They are actually the leaves of the Pandanus species are actually fourfold. So based on this example, you have here 1, 2, 3, 4. So there are actually, so if you imagine a paper, if you fold the paper into half, and then each half you fold it again into another half, so you now have 4 quadrants. So that's how the leaves of a Pandanus species would look like. You have 4 quadrants in a Pandanus species leaf. So it's very much easy to locate them in the field. Okay, now So the studies of the Pandanus alkaloids are actually highlighted or elaborated in two major publications. So it's actually a compilation of the studies, both the phytochemical studies and synthetic studies. They are published in the Alkaloids, which is a well-known book series about the alkaloids. So the first publication about the Pandanus alkaloids was in 2008, wherein in the 2008 it covers the it covers the studies on the Pandanus alkaloids starting from the early 90s up to 2008, and then we were invited again through my professor, Professor Hiromitsu Takayama from Chiba University. We were invited again to write a book, a chapter, I mean, about the alkaloids, about the recent progress in the chemistry of the Pandanus alkaloids, which is published in 2019. So here are the two major publications, or the highlights, the compilation of the synthetic and phytochemical studies about the Pandanus alkaloids in the alkaloids. So there are important people here. So Maridal Nonato is actually Professor Nonato is actually my advisor in my bachelor and in my master's. So she's the one who introduced me into the wonderful world of the Pandanus species. And of course, Hiromitsu Takayama is my supervisor during my PhD. So Professor Takayama, his research interest is actually on the alkaloids from various plant species, in particular Rubiaceae, Pandanus, and then other species focusing on the alkaloids, of course. And then the other one would be Mary Garson. Mary Garson is the advisor of Dr. Nonato in I think in Australia. So in Wollongong University. So these are the major highlights of the studies on the Pandanus species. Now, so in terms of the extraction, there are two methods on how to extract the initial extraction of the Pandanus species or the Pandanus alkaloids. So one of them is that extraction by polarity. So what do we mean by polarity? So basically, the leaves, the air-dried leaves are extracted with methanol, and then the methanol would be separated according to polarity using hexane, and then a semipolar solvent such as chloroform or dichloromethane or ethyl acetate, and then the last would be a more polar solvent such as butanol. Or other than bipolarity, sequential extraction is also being used. So what do we mean by sequential extraction? You have the air-dried ground leaves, and then first you extract the leaves directly with a non-polar solvent such as hexane or an ether in order to remove the fats or any garbage. And then we extract, and then after drying again, we extract that with the semipolar such as dichloromethane or chloroform or ethyl acetate, and that is where most of the alkaloids are soluble. And I mean, most of the Pandanus alkaloids are soluble. And aside from extraction according to polarity, the crude extract that we have is also the easiest way is also to separate the alkaloid fraction, what we call the crude base fraction, using the acid base extraction. However, there are studies being conducted on the Pandanus alkaloids wherein when we try to use the acid base extraction, it may produce some artifacts. So which means that because we are using an acid to initially separate, okay, since we're using an acid to initially separate these alkaloids, some reactions may occur, some acid catalyzed reactions may occur, and hence producing some artifacts. Now, so what are now? So initially, for this one, I am going to highlight the the first studies conducted on the Pandanus alkaloids, or the first isolation of Pandanus alkaloids. So these alkaloids were isolated from the Philippine Pandanus amarillifolius. So initially, or rather, based on the literature, the main bulk of studies on the Pandanus alkaloids are focused on the Pandanus amarillifolius. Again, the Pandanus amarillifolius, these are, you can actually distinguish Pandanus amarillifolius because it has a distinct smell. And that smell is, was identified as 2-acetyl pyrrolene. Now, so the first isolated alkaloid from Pandanus amarillifolius, or what we call in the Philippines pandan mabango, would this pandamarine. As you can see in the pandamarine, it has a spirocyclic ring, a lactam ring. So you have here 2 lactam rings. So it was pandamarine was crystalline in structure. So it was first isolated first published in 1992, and after that, pandamarine has never been isolated before. After 1992, pandamarine has never been isolated again. So the story here is that Dr. Nonato told me that in this case for pandamarine, so they left an extract of the Pandanus amarillifolius, and then after some time they were able to observe some crystals. So the crystals were separated, and then what is also an important story about the pandamarine is that the structure was not determined by spectroscopy, but the structure of pandamarine was determined by X-ray crystallography. Okay, so pandamarine, the main structure, or rather the structure, was actually determined by X-ray crystallography in 1992. And then we now have here the pandamarylactone 1, pandamarylactone 32, and the pandamarylactone 31. These 3 alkaloids were isolated by Dr. Maribel Donato, by Professor Donato, during her PhD in Australia at the University of Wollongong. So these 3 alkaloids were published in 1993. As you can see here, pandamarylactone 1 would have a similar structure with that of pandamarine. The only difference is that pandamarine would have a lactam structure, while pandamarylactone 1 would have a lactone structure, wherein this lactone structure were also elaborated in pandamarylactone 32 and pandamarylactone 31. And as you can see, as you are going to see later, that this feature, okay, this butylidine alpha beta saturated this unsaturated structure here in the Pandanus alkaloids are actually common in most of these Pandanus alkaloids, or Pandanus amarillifolius alkaloids. And then from Thailand, okay, so Pandanus amarillifolius, which were bought or collected from Thailand, known as Toy Hom. So there are a variety of alkaloids. You have here the pandamarylactone A, pandamarylactone B, pandamarylactone C, and pandamarylactone D. So actually, pandamarylactone A and pandamarylactone B are actually A, B, C, and D are diastereomeric compounds. So A and B are actually enantiomers. So they differ on the stereochemistry at this the methane carbons. They differ on the stereochemistry at these methane carbons, wherein for pandamarylactone A, it has a trio configuration, while pandamarylactone B would have an erythro configuration. And then this stereochemistry here were also identified from pandamarylactone C, and D, wherein C and A would have the same stereochemistry, and B and D would also have the same stereochemistry. The main difference between AB with C and D is that the orientation of the double bond in the in this butylidene structure. So in A and B, the double bond would have a Z configuration, while in the C and D would have an E configuration. Now, also from Thailand, Pandanus amarylifolius, norpandamarylactonin A and norpandamarylactonin B were also isolated. Now, an important alkaloid which was isolated back then was this pandanamine. Why is this important? Why pandanamine is very important? Because it is known that pandanamine, based on the biogenetic consideration of the Pandanus alkaloids, the pandanamine is said to be a precursor. So that is why the isolation of the pandanamine would represent, a major breakthrough in the Pandanus alkaloids chemistry, because this is a proposed precursor in the biogenesis of all of these in some of these Pandanus alkaloids. Now, of course, in Indonesia, I hope the name of the Pandanus amarylifolius is correct. I'm not sure if Pandanus amarylifolius in Indonesia is called daun pandan. I hope it's correct, no? So from the Indonesian, is it correct? Yeah, it's correct. I hope so. Yeah. So in Indonesia, or the daun pandan sample, the Pandanus amarylifolius sample, so we have here the first isolated alkaloids from these Indonesian samples would be the pandamarylactam 3Y and 3X. So basically, they have the same structure. They just differ in the orientation again. Okay, they differ again in the orientation of the alkene double bond in the upper part of these alkaloids, wherein the pandamarylactam 3Y would have the Z configuration, and the pandamarylactam 3X would have the E configuration. And then in 2004, from the laboratory of Dr. Professor Medigarson in Australia, they were able to isolate mixtures of the pandamarylactonins A, B, and C, D, and then also they were able to isolate an oxidized product of the pandamarylactonin. So here you have an N oxide. So as you can see, this structure is actually the pandamarylactonins A to D. Now, so I didn't specify here anymore the stereochemistry, because it may represent a mixture, So well, the important thing is here is that they were able to isolate an N oxide, or the oxidized form of this pandanus, or rather these pandamarylactonins. And as well, they were able to isolate the E configuration of the pandanamine. So again, the pandanamine would have a Z configuration, while for the Indonesian sample, they were able to isolate the E configured pandanamine. Now, this one, they didn't give a special name for this alkaloid. So, based on the study on this Indonesian pandan, so when they compared the crude extract before and after acid-base extraction, they were not able to identify this peak, this alkaloid peak in the crude. Now, this peak was only made visible after acid-base extraction. So they propose that this alkaloid is an artifact isolated because of an acid-base extraction procedure. So So those are the one that I showed you, the pandamarilactones, the pandamarine, the pandanamine, the pandamarilactam 3X and 3Y are the first alkaloids which were isolated all from Pandanus amaryllifolia coming from different sources in the Philippines, in Indonesia, and in Thailand. And that in terms of their biogenetic origin, it was proposed that the Pandanus alkaloids originated from leucine and from the glutamate. So from leucine, it would produce molecule A, and from glutamate would produce molecule B, and condensation of A and B would produce molecule C, and then from molecule C would, based on some reactions such as the carboxylation, the amination, reduction, it would eventually produce pandamarilactone 32 and 31. Now, so here would again produce this particular precursor, which may eventually produce pandamarilactam 3X and 3Y. Now, C would undergo a series of reactions such as the carboxylation, such as reduction, dehydration, or deamination, may produce either the pandamarine or, as you can see here, the pandanamine. Take note that among the precursors, the pandanamine is the only precursor which was isolated from the Pandanus amaryllifolia. And eventually, this pandanamine would give rise to the pandamarilactonines and the pandamarilactone 1. And eventually, for this molecule, might be a precursor for the pandamarine. Now, so based on the alkaloids which I showed you, we as you notice, we have this different or similar structural features of the Pandanus alkaloids. So the Pandanus alkaloids are known to possess a gamma butylidine, alpha methyl, alpha beta, and saturated lactone, which is elaborated by the pandamarilactonines, and then the pandamarilactones in the upper part. Also, we have the Pandanus alkaloids would also have a gamma butylidine, alpha methyl, alpha beta, and saturated lactam, which is elaborated by your pandamarine, by the pandamarine, which is the very first alkaloid isolated. Now, the Pandanus alkaloids are also known to possess a pyrrolidinyl, alpha methyl, alpha beta, unsaturated lactone, wherein in this case it was elaborated by this structure. You know, the pyrrolidinyl, you have an alpha methyl, alpha beta, unsaturated butyrolactone moiety. And of course, the pandamarilactones 132 would have a piperidine spirocyclic moiety, or rather the pandamarine would also have the piperidine spirocyclic structure. So these are, the common structural features in these Pandanus alkaloids. Now, so again, from the Pandanus amaryllifolia, which were collected from the Philippines. So since the Pandanus alkaloids, the isolation of Pandanus alkaloids in Pandanus amaryllifolia were concentrated on the leaves, so we try also to investigate what could be, or rather are there alkaloids which can be found in the roots. So this is my initial work on the alkaloids from the Pandanus, from the Pandanus alkaloids. So we have here the roots which were extracted with methanol to come up with the methanol extract. And then using acid-base extraction, we were able to separate the crude base, and then the crude base would have, all the alkaloids, or rather the crude base is the alkaloid fraction. So we were able to separate the alkaloid fraction from the non-alkaloid fraction. The ethyl acetate, or rather, the ethyl acetate layer would contain the non-alkaloidal compounds. So the crude base, which is about 2 grams, was separated by using various chromatographic methods. So I normally use medium pace lipid chromatography or aminosilica gel chromatography. So using aminosilica gel is very much essential, or rather an advantage when you are isolating actually the alkaloids. But because all the garbage, if there are non-alkaloids in your extract, they will not be eluted if you have the amino acid silica gel. So from this crude base of the Pandanus amarylliferous roots, we were able to identify 5 new alkaloids in addition to the known alkaloids, previously known alkaloids. Now, so what are these alkaloids? So we have here pandamarilactonin E, pandamarilactonin G, we have pandamarilactonin F, pandamarilactonin FN oxide, and pandamarilactonin H. Now, so in terms of the structure, as you can see here, pandamarilactonin E, F, and FN oxide are just the reduced product of the pandamarilactonins. Okay, so we remove here the double bond, we remove here the double bond, we remove here the double bond, making now these compounds a little bit complicated. Why are they now complicated? Because of the removal of the double bond, we now have a stereocenter. So when you're doing isolation or phytochemical studies, in particular, if we are able to isolate a new compound, you have to identify this. Very important that if you want it to be published in a good journal, it is very essential that at least you identify or you have an idea of the absolute stereochem or relative or if you can, the absolute stereochemistry of these stereocenters. Now, so the pandamarilactonin E, pandamarilactonin G, and the pandamarilactonin H, we were able to identify the stereochemistry, the absolute stereochemistry by total synthesis. While for pandamarilactonin F and oxide, we were able to identify this by subjecting both pandamarilactonin E and pandamarilactonin F via oxidation using MCPBA. Okay, so in MCPBA, the mechanism there is that we try to oxidize the nitrogen atom to have like a nitrogen double bond O, so to produce a quaternary nitrogen atom. So in this case, we compared the oxidized product of the pandamarilactonin E and the pandamarilactonin F, and the how should I say, the NMR spectra is the same as that of the the product coming from pandamarilactonin F. Therefore, this is now known as pandamarilactonin F N oxide. So this is what happened here. Now, also, in this one, this is also the first isolation of a Pandanus alkaloid bearing this structure here. So we have here, this is the most common structural feature wherein you have pyrrolidine ring and a butyrolactone structure. However, in this case, we have a pyrrolidine ring and a methyl acetate structure, which is the first structure reported in the Pandanus alkaloids. Now, so this is now the synthesis of the pandamarilactonin G. Pandamarilactonin G, of course, this butyrolactone alcohol, which was previously I previously synthesized, when I synthesize dobusamine A, where I'm going to show you later what is dobusamine A. So this butyrolactone alcohol was converted to a butyrolactone iodide, and it was condensed with pyrrolidone to yield now the pandamarylactonin G. While for the pandamarylactonin E, the synthesized, so the dobusamine B, where I'm going to show you later what's a dobusamine B. Dobusamine B, wherein it has this double bond, was subjected to a hydrogenation reaction using hydrogen gas in 10% palladium and carbon, would now yield the synthetic pandamarylactonin E, wherein the NMR data is exactly similar with that of the natural product. Now, for the pandamarylactonin H, I think I was not able to, well, I was not able to put here the synthetic scheme for pandamarylactonin H. However, for the pandamarylactonin H, the absolute stereochemistry was determined using diproline, wherein the diproline would have an R configuration, and then we were able to synthesize this positive pandamarylactonin H. However, when we compare the optical rotation of the synthesized pandamarylactonin H, it has a positive optical rotation, while the natural product would have a negative optical rotation. Therefore, we conclude that the natural product would have an S configuration at this chiral center. Now, you might be asking, why did we use a diproline wherein the diproline would have an R configuration? Because the main reason for this one is that the known alkaloids, in particular the pandamarylactonin A, wherein the pandamarylactonin A would have, established synthetic studies, would have an R configuration at this chiral center. So basically, we initially assume that pandamarylactonin H would have a similar absolute stereochemistry with the known Pandanus alcohols. But eventually, when we compare the optical rotation is the opposite. So we concluded that it has, a 14S configuration other than the normal 14R configuration of from the previously reported Pandanus alkaloids. And in this case, this is the proposed biogenesis of the pandamarylactonin H. The pandamarylactonin H was proposed to originate from glutamate. The glutamate would undergo a decarboxylation to give rise to an a gamma aminobutyric acid, or GABA, and to yield this and then this GABA would undergo a condensation with the same molecule to yield the symmetrical structure. And also from the leucine would undergo a series of reactions to yield this particular precursor. And a condensation of these two would yield this precursor here. And via a manic reaction or a series of reactions, deamination, decarboxylation, dehydroxylation, etc., would now give rise to an iminium structure, a very important iminium structure. And the acetyl group here, the acetyl group of the pandamarylactonin H, so you have here an iminium ion reacting with S-methyl methionate or a methyl acetate, for in this case, to now have the pandamarylactonin H. Okay. Now, so aside from here, so let me finish. So this one now. So aside from the Philippine sample of Pandanus amarylifolius, from the Indonesian sample of Pandanus amarylifolius, from the Thailand sample of Pandanus amarylifolius, researchers from Taiwan, in collaboration with Dr. Maribel Nonato, they were able to investigate, the Pandanus amarylifolius being grown or rather collected from Taiwan. And in this case, they were able to isolate 16 new alkaloids, which would include the pandamorilactone 2, 3, pandanmenyamine, pandalactonine, which are actually the analogs. Okay, as you can see, based on this structure, you have this alkylidine, alpha methyl, alpha beta unsaturated butyrolactone. This is from pandamorilactonine. They just differ in this configuration. So these are actually the analogs of the previous Pandanus alkaloids. However, what is essential here would be the isolation of this pandanmenyamine, which is the first one to have an amide. So this one would have an open amide structure, and also the pandamorilactone 3 would now have again a spirocyclic lactam structure at the lower part. Remember that we first encountered this structure in pandamarine. The pandamarine, again, is the first isolated Pandanus alkaloid. And of course, the pandalactonine would just be presumed to be originated from the hydroxylation, say for example, of a pandamorilactone, and via a cyclization reaction would now have the pandalactonine. Now, what is the highlight of the Pandanus amarillifolius collected in Taiwan is the isolation of these new structures, the pandalizine A, B, C, D, and E. As you can see, the pandalizine A, B, C, and D would now have a new structure which is different from the previous Pandanus alkaloids. So pandalizine A, B, and C, D, and D, A, B, C, D, and E would now have the indolizine moiety, which is not encountered before from the Pandanus, from the previous Pandanus alkaloids. So this is, the proposed genetic precursor, proposed genetic, not precursor, but the proposed biogenetic consideration of this new Pandanus alkaloids collected from Taiwan. So from I think this is hydroxyl glutamic acid. When you react it with GABA, which would produce this precursor, and via a series of reaction may yield the pandalizines A to E. And then again, using the same molecule, which is methyl, I think this is methyl glutamic acid, reacting with this molecule would eventually produce the pandanmenyamine, and then the N-acetyl norpandamorilactonines A and B coming from the pandanmenyamine. And eventually, this symmetrical structure here would undergo a series of reaction to now yield the pandalactonine. So this is now the proposed biogenesis of these Pandanus alkaloids from Taiwan. Now, since the Pandanus alkaloids are all based on the previous studies, are all isolated from the same species of Pandanus, we also try to investigate other Pandanus species which may have, the presence of the alkaloids. And one of them, so this is another work of mine, Pandanus dubius. So Pandanus dubius, in this case, Pandanus dubius, leaves of Pandanus dubius were used. So air-dried ground leaves of Pandanus dubius was subjected to methanol extraction to yield now the methanol extract. And then the methanol extract was then subjected to an acid-base extraction to yield now the crude alkaloid mixture, and via a chromatographic separation, we were able to obtain 2 new alkaloids in addition to, you know, the previously known alkaloids. And these 2 alkaloids coming from Pandanus dubius are the dubisamine A and dubisamine B. I am going to explain later why do we have a dubisamine C. Okay, so why is it named dubisamine? Dubisamine because we name it after, you know, after the Pandanus species that we use, which is Pandanus dubius. And normally, if you want to name a new compound, we usually name it using the name of the plant, and then since this is an alkaloid, of course is an amine, so the name would end with amine. So we name this as dubisamine. A and B are the new alkaloids from Pandanus dubius. As you can see here, the dubisamine B, the structure of dubisamine B is actually the same as that of the previously isolated pandamarylactonines. What is the difference? The difference is that we just have here the removal. Okay, so we have here the removal of the double bonds here in this position and in this position here. So again, since we now have a chiral center, we have to identify the absolute stereochemistry of these chiral centers. And the chiral center, the absolute stereochemistry of the dubisamine B was determined by a total synthesis. And the same is true for dubisamine A. As you can see here, for dubisamine A, the structure is similar to that of the pandanamine. Okay, so pandanamine, it is the symmetrical Pandanus alkaloid having the 2 double bonds. So here we just remove the double bonds from the pandanamine, and then we now have the dubisamine, or rather the double bond of the pandanamine was reduced, and we now have here the dubisamine A. So this slide would now elaborate the total synthesis of dubisamine A. So dubisamine A, so we use an inexpensive and not so very much cheap material, a 1,6-hexane diol, which underwent protection and oxidation to come up with this aldehyde. And then this aldehyde was subjected to a proline catalyzed the amino oxidation reaction to produce this particular alkene alcohol, wherein this alkene alcohol is now the source of the stereochemistry of the butyrolactone of would now be the source of the chiral carbon of this particular methine carbon here. So based on HPLC, this particular molecule would have a 99% enantiomeric excess. And via a series of reaction, which would include lactonization reaction and Mitsunobu reaction, a series of Mitsunobu reactions. So this is a Mitsunobu reaction here. And then a deprotection of the removal of the protecting agent of the nitrogen atom, we were now able to synthesize in the enantiomerically pure dubisamine A. So this is now the proton NMR spectra comparison of the proton NMR spectra of the synthetic dubisamine A with that of the natural product dubisamine A. Now, so what you can see here, what we notice here is that in the natural dubisamine here, dubisamine A, we have this additional peaks at around 4.3 ppm and at around 2.5 ppm, wherein we assume that this particular peaks here, we assume that this is the diastereomer of the dubisamine A. So take note that for the dubisamine A, we have here an anti-configuration, okay? So the methyne protons here in this carbon and in this carbon would have an opposite orientation. Okay, so there is an opposite orientation of the methyne carbon, the CH here, and the CH here, the hydrogen here, and then the hydrogen here would have an opposite orientation. So we assume that these peaks that we can see here would be the syn. So here in the natural dubisamin A, we have an anti-configuration between the two stereocenters. So we assume that these peaks here might be due to the syn configuration of dubisamin A. And to prove that, we were able to synthesize the minor, the plausible minor alkaloid of dubisamin A, which we named now as dubisamin C. So we were able to do a total synthesis. However, this is not an enantiomer synthesis. This is not we were not able to determine anymore the absolute configuration. What we just did here is just to prove that the methane would have a syn configuration, or the orientation of the hydrogen atoms are on the same side. So basically, the synthesis would commence using again a very cheap molecule, a 1,5-pentanediol, which is subjected to a protection of one of the hydroxyl groups, an oxidation, and a Grignard reaction to produce this particular molecule. And via a ring closing metathesis using a Grubbs catalyst and a hydrogenation reaction, we come up now with compound 77. This 77 was subjected twice to a Mitsunobu reaction. So using PPH3 and DAD would give you a Mitsunobu reaction to come up with the symmetrical protected amine. Now, the nosyl group in the amine was again removed using potassium carbonate in the presence of phenol sulfide to produce now dubisamin C. So this is now a comparison, a comparison now of the NMR data of the synthetic dubisamin A, the synthetic dubisamin C, and the natural dubisamin A, As you can see, our hypothesis, our hypothesis that the minor peaks of the natural dubisamin A would now correspond to the minor compound or the minor alkaloid is actually true, because here, so the peaks here coincide now with that of the synthetic dubisamin C. So that is why in this particular slide, for the Pandanus dubius, aside from dubisamin A, we were able to identify, based on synthesis and comparison of the NMR data, a minor alkaloid, a potential minor alkaloid, which is dubisamin C. Now, I think this is now my last slide. So another Pandanus species, which was determined to have the presence of the alkaloid, would be the Pandanus utilis. The Pandanus utilis were collected from Egypt, and that this one, from the Pandanus utilis, two new alkaloids again were isolated, Pandalicin A and B, wherein the structure, or rather the key structural features for this Pandalicin A and B were similar to the previously known, again, Pandanus alkaloid. So here, again, you have the indolizidine ring, and then you have here the alpha-methyl, alpha-beta unsaturated butyrolactone moiety. What is different? What is different between the two is that they differ in the configuration or the orientation of the double bond here. So here you have a Z configuration, while here you have an E one E configuration, and the other one would have a Z configuration. So this is now the proposed biogenetic pathway for the pandalizine A and B from the gamma aminobutyric acid and leucine, producing these compounds, and condensation of these two compounds and a series of reaction would give you this particular potential precursor, and this potential precursor will give you an iminium ion, and via a series of reaction would eventually produce the pandalizine A and B. Now, so in conclusion, so again, as you can see here, out of the how many? Out of the 700 species, no? Out of the 700 species Pandanus, out of the 700 Pandanus species, only 3 so far, okay, only 3 so far were found to contain alkaloids, and this would include the Pandanus amarylifolius, Pandanus utilis, and Pandanus dubius. And the structural features of these isolated alkaloids would contain an alpha butylidine, alpha methyl, alpha beta unsaturated gamma lactam structure, a gamma butylidine, alpha methyl, alpha beta unsaturated gamma lactone structure. We also have the Pandanus alkaloids. Also present would be the pyrrolidinil, substituted alpha beta unsaturated gamma lactone structure. And of course, for the pandalizines, we have the indolizidine core structure or moiety. So again, before I end, I would like to, acknowledge the 2 important people who introduced me, very helpful in my career in terms of this Pandanus research. So to Professor, Maribel Donato, she's my mentor in my bachelor's and in my master's, and she introduced me to this world of Pandanus. So Dr. Maribel Donato's work is actually focused on this Pandanus species. And of course, my PhD supervisor, Professor Takayama. So Professor Takayama, his interest is on the alkaloids. So that is why my interest on this Pandanus alkaloids were also enhanced because of Professor Takayama. And with that, thank you so much for listening. Thank you.
Thank you so much, Professor Tan, for your sharing knowledge to us. It was really great presentation. There are so many information here. I hope that all students and participants here can receive something and give questions, because now we are going to have discussion. So, but first of all, I would like to ask something, Professor Tan, before participants here raise their hand and give questions. It is interesting that only from Pandanus species with particular structure of alkaloids, they gave lots of variety of the structures. There are a variety of structures from Pandanus amaryllis species with 3 different geographic areas. They gave different structures of Pandanus alkaloids. It seems that I don't know whether so I would like to hear your opinion. It seems that this geographic really affect to the structure of Pandanus alkaloids. Is it correct?
Yes, actually, yeah, Dr. Rini, you are actually correct, because the natural products being isolated from a particular plant is actually dependent on its biographical location. It is also dependent, on the pH of the soil, on the environment, etc. However, let me point this out that based on my current study, so I actually repeated again, the isolation of Pandanus of alkaloids from Pandan amarylifolius. So I actually did that. So I was able to have a fellowship from the JSPS. So I spent like last year, I went again in Chiba University, but with a different professor. I restudied again Pandanus amarylifolius. And what I found out is that the Pandanus, that particular Pandanus amarylifolius, which we collected here in the Philippines, I was now able to get all of these varieties of structure. So I was able to get these pyrrolidine structures, this piperidine, including this indolizine. So yeah, so in a way, yes, it is true that biographic location is quite an important factor on what are the natural products, but it might also be that it was not previously isolated because perhaps these alkaloids are minor. So if you remember that natural products in plants are quite small in amount. So if they're very
Yes.
small, we might be missing them. So it could be possible that they are actually present, but they are in minor quantities only. However, you are correct that biographical location would have a great effect on in particular, the alkaloids being isolated from plants.
Yeah.
Mm.
So now we have questions here from. Oh, excuse me. Wait a second. First from Anissa Fernanda. Anissa, I think you can ask directly to Professor Tan. What about that? Where is Anissa? Anissa, are you here?
Yes, ma'am. I'm here.
So,
Yeah. So you wanna to asking it's sir, right?
Yes,
Okay,
hello.
sir. Okay. Maybe my question is out of the topic, but um, I think I'm feeling good when you can to answer my questions. So, good morning on Indonesian. Uh, I just want to asking, does the Philippines use pandan for handicrafts like a bag, ring, or etc.? I just want to know.
Yes, actually, not actually Pandanus amarylifolius per se, but a different species of Pandanus. So here in the Philippines, there is a place known as Cavinti in Laguna, where in that particular place in the Philippines, in Cavinti, Laguna, they cultivate a Pandanus species known as Pandanus simplex. So that Pandanus simplex is being used in handicrafts, in making bags, in making mats, etc. So not actually the Pandanus amarylifolius, but other Pandanus species, because the leaves of Pandanus of this some of these Pandanus species are actually the leaves are really hard and fibrous, and it's very their main application in the small industries here in the provinces, they're being used to make some of these handicrafts. Yeah. So they're quite very much useful in some other parts here in the Philippines as well.
yes, I remember that I received your gift from Pandanus leaf, actually, Professor Tan.
Yeah.
Is it the basket that then I use it
Oh, really?
almost every time when I went to the market.
Okay, yeah.
It is still here.
Oh, yeah.
So thank you, Anissa, for the question. So yeah, it is not Pandanus amaryllis, but Pandanus simplex.
Okay, I
All
think
right, Ersando already raised his hand. So please, Ersando.
Okay. Is my voice heard clearly, Mrs.?
Yes, yes, you're here clearly.
Okay. Thank you, moderator, Mrs. Rini Muharini, for giving me opportunity. Hello, Professor. Great to meet you today.
Hello, yeah, nice to meet you.
I'm really excited about natural chemistry, and so today I want to ask. But before I ask my main question, I have first question for beginning my main question. Are all the alkaloid that you described is potentially neuroprotective?
Oh, okay. Now, that is what I am trying to investigate now in my research. As because there are no or rather there is quite a limited study on this Pandanus alkaloid. So basically, the previous studies conducted on the Pandanus alkaloids are on just in their chemistry because of their unique structure. Scientists all over the world are being interested in terms of their synthesis. So there's no study conducted yet, or there's one. So we made I think that was in 2014 or 2015, we were able to do an antibacterial activity of the major alkaloids, the pandamarylactonines. Aside from that, there's no other biological studies yet. So that is what I am currently doing now. So I started doing neuroprotective studies on the other Pandanus species, including Pandanus cardinensis, Pandanus sectorius, etc. And then we try to associate, the neuroprotective effect of some of these Pandanus species, not with the alkaloids, but other compounds, such as the sterols, because these Pandanus species, the ones that I studied, do not contain any alkaloids. Now, in Pandanus amarylifolius, I was able to do, based on my latest paper, I was able to do, the neuroprotective effect on this crude alkaloid extract. So I was able to separate the crude alkaloid extract, and it has a very promising activity. Now, I was able to isolate, so it is not reported here because I did not report here because we have not published yet, but we are going to report potentially new alkaloids. Now, these new alkaloids and the known alkaloids, we just have to test them first. Uh, um, it will be subjected to neuroprotective test because neuroprotective test would be a little bit tedious, so it's time consuming. And also, however, we associate one of the compounds. So it is very much interesting that one of the compounds that we identified in my latest work in Pandanus amarylifolius is nicotinamide. Okay, so nicotinamide is we were able to identify nicotinamide from the crude base of Pandanus amarylifolius. And what is good about nicotinamide is that it has a clinical trial as an Alzheimer's anti-Alzheimer's drug. So initially, we tried to associate the activity of the Pandanus amarylifolius crude alkaloid extract to nicotinamide, but we are currently doing now the assay on the major alkaloids from Pandan, because in this neuroprotective assay we have various assays. We also have, we should also have a sufficient amount. As you know, the natural products that you isolate sometimes is not sufficient for biological assays. That is why chemical synthesis as well is very much important in natural products. Remember that in biology, if you want to test the biological assay, it is the your main consideration here would be the amount plus the purity of the natural product. Okay, so yeah, so as of now, only the crude alkaloid extract would have a neuroprotective activity, and we associate that might be due to the nicotinamide, which we also isolated from the crude alkaloid extract.
Okay, thank you, Professor. Maybe I got it what you mean. And can I continue to my main question?
Sure, yeah.
Okay, so my main question is what are the methods or steps for the initial analysis to find that plant has potential to be a new protector? Can it be directly tested from its extract with a specific test, for example, new protective assay, or must it be based on previous information obtained from other people that this plant is useful for that? I mean, for new protective or something else?
Okay. Yeah. So um when you talk about neuroprotective effect, you are trying to protect something. And in this case, in my assay, um, um, my neuroprotective study is cell-based. So I am using neuroblastoma cell. So I am using an SHSY 5Y neuroblastoma cells. Okay. So when we talk about neuroprotective assay, um, let me just run down, let me just give you the step. So when you now have the cells, um, in Alzheimer's disease, there are different, or rather the main peptide associated with Alzheimer's disease is amyloid beta. Okay, so amyloid beta 40 or 42. And that this amyloid beta by itself at a certain concentration is toxic to the neuroblastoma cells. Okay? So my objective is to treat the cell with the crude extract, any plant extract, or the compound, and I treat that with the amyloid beta. If the amyloid beta did not produce any toxicity with the cell, then we can say that the particular extract or compound has a neuroprotective effect because by itself, the amyloid beta is toxic to the cell. So we are trying, when we talk about the neuroprotective assay, neuroprotective effect, we are trying now to monitor the viability of the cell against its toxicity with the amyloid beta. Can the compound or the plant protect the cell? It can be answered if there is a significant increase in terms of the cell viability. That's how we do neuroprotective assay. Now, in your question, is there an initial method in order to know? There is an assay known as the THT, the thioflavin T assay. The mechanism of the thioflavin T assay is that the extract or the compound, you can determine whether the extract or the compound will inhibit the aggregation of the amyloid beta, because the amyloid beta will only be toxic to the cell or to our brain if it forms a nucleation, if it aggregates, if it forms plaques. So that would be an initial step in order for you to determine, if you don't have any cell assay, that would be a first step, in my opinion, to determine whether a particular compound or extract might have a neuroprotective effect, the thioflavin assay. Because the thioflavin, it's just uh, would only use the thioflavin molecule and then the amyloid beta. It would not use any cells, so you can actually use that for you to determine initially if the plant or the compound could be subjected to a neuroprotective assay. Okay.
Oh, okay, Professor. I got it.
Okay, thank you.
Maybe
Thank,
just only that my question, and I give it back to the moderator, Mrs. Muharini. Thank you very much for the opportunity.
Thank you.
thank you, Ersando. Now we have Putri Risti Hidayati, who already raised her hand. So, Putri, you can activate your camera and give question to Professor Tan.
Thank you for Bu Rini. So I would like to ask about, in terms of the number of alkaloids in it, and the Pandanus alkaloids, would the isolation or extraction process affect the potential or quality of the alkaloids? Like, it goes through a heating process, then would it make it less superior or make it more superior if it's compared to the one that hasn't been isolated?
I'm sorry. Can you please again repeat the question? I was not able to get it clearly.
I'm asking about the process. If it's going through a process like isolation or extraction that may be going through a heating process, would the
I'm sorry. Healing. Sorry.
Heating.
Heating. Okay. Heating. Okay.
Yes.
Reflux. Reflux.
or the potential of alkaloids become more or less superior?
Became more or less real.
Superior.
Superior. I mean, she
If it's
meant that whether with the reflux or socculation make the structure of alkaloids become composed, decompose, or something like that.
Or okay. Yeah.
Yes. Yeah.
Okay. Thank you so much for that question. Well, when I was still in Japan, well, in the Philippines, we don't extract via a reflux method. Okay. So in Japan, since the plant that they use there, because they don't have this, unlike here us in Indonesia, in the Philippines, in Malaysia, we have a biodiversity, a very good biodiversity. So in my experience in Japan with my lab mates before, since they only have a small amount of the material, they usually extract the plant via reflux. And then in terms of the reflux extraction, they only subject that to like 30 minutes. What is good about the reflux method is that since you are heating, the time of extraction is also lessened. However, you have to take note that since also we are heating, it is possible, yes, it is possible that we may produce artifacts when we do reflux method. For the Pandanus alkaloids, as I mentioned in my presentation, using acid-base extraction might, based on the work of Dr. Mary Garson in 2004, they were able to isolate an artifact. Okay, so yeah, a reflux method, and in our case, the acid-base extraction method, it is possible that we are producing, that it might produce artifacts. So what we do here is that we try to compare, the LCMS data of our isolated compound with that of the crude extract. So when we talk about the crude extract, that is when we try to extract the plant initially, the air dried ground leaves with the with either methanol or ethanol, and then if the peak is still the alkaloid peak is still present in the initial crude extract, then we are sure that it is indeed a natural product. However, if we cannot see that peak anymore, or if we try to compare, say for example, a crude alkaloid extract with that of the crude alcohol extract, if there is an additional peak in the crude alkaloid extract, then it might be that that particular peak may be an artifact due to, might be due to the extraction process. Yeah. Okay.
Okay, I think I got it now. Thank you so much.
Thank you.
Okay, thank you, Putri. So, Professor Tan, which method do you prefer? Whether the classic maceration or reflux for alkaloids?
For alkaloids, well, some alkaloids are actually stable. So if you want to do, if you want to do a reflux method, well, case to case basis. If you have a small amount of sample, then reflux method would be
Reflux method, and then
because you
yeah.
know, if you are going to do the conventional method, at some point you also you just the yield of the crude extract is less. Unlike if it is a reflux
Yeah.
method, you are sure that you're going to get more of the extract. Okay, so it's a case to case basis, and then also you also have to study. I think you also have to study what are the typical compounds that you can isolate on a particular plant species. If you see that these compounds can easily be manipulated, when I say manipulated, or can easily be destroyed, or can easily be decomposed, or if a particular plant species would have a lot of hydroxyl or phenolic or polyphenols, doing reflux method might not be the good method, because you know there might be methylation happening, etc. So it's a case to case basis. But personally, I do prefer I always use the conventional uh method, using the percolation method. Yeah.
Percolation, yeah.
Yes,
Perhaps
percolate.
we can use sonicators.
Uh-huh.
Sonicators.
Yes,
You know,
yeah.
there is a ultrasonic
Yes,
method that can shake this membrane cell in the organism
yes,
plant.
yes. Actually,
Yeah.
so the sonication is also very important if you have a small amount, because in our case we have like kilograms, you know, so we collect
Yeah.
kilograms of the plant. So we use the percolator, but we only have, if you only have, say for example, if you only have 100 grams, then we try to sonicate that one. Yeah, in order for
Yeah.
us to
So
have a good yield of the extract.
that's true. What we did here, we normally use a conventional method, maceration,
Yeah.
and we only got less than 10% for its
Okay.
rendement. So it's really, yeah, that's why we are seeking now what method that can produce more than 10%.
Yeah, actually, yes, yes. That is actually, you know, one of the disadvantages if you're doing a natural products chemistry, because the yield is really small. Yeah.
Yeah, that's right. All right. We move to another question. Here is from Ahmad. I think Ahmad also can give question directly to Professor Tan. Ahmad, are you still here? Oh, he's gone. All right, we move to another question from Dea Aninda. Aninda, you can also give directly to Professor Tan.
Yes, ma'am. Wait. Okay, thanks for opportunity, ma'am. I want to ask, based on the journals I've read, the alkaloid content of Pandanus that grows in each region is different. Now, how does the type of soil in each different region affect the alkaloid content in Pandanus? For example, in Pandanus that grows in Thailand, Indonesia, or Philippines, as explained by Mr. Tan earlier, and which type of Pandanus grows in which area has the highest alkaloid content among the three countries? Thank you.
Okay. As I know, based on the isolation of the alkaloids, in the case in previously, no, so previously under Professor Takayama, they were targeting the major alkaloids. Okay, um so in terms of the major alkaloids, it would be the pandamrylactonins. Um And based on what we know, so from the Philippine samples, from the Thailand samples, and the majority of the alkaloid that we were able to isolate would be the pandamarylactonines. Now, for the samples in Taiwan, as you can see, the alkaloids isolated were just analogs of the pandamarylactonines. Now, in terms of the biographical location, as we have mentioned a while ago, yeah, the pH of the soil would have a great effect. You know, the climate as well would have an effect. So apparently, there's no study yet. So there's no study yet on the effect of the pH of the soil or the climate as regards to the type of alkaloids or what are the major alkaloids being isolated, because the phytochemical studies on these alkaloids are only focused on, you know, the isolation of alkaloids. So perhaps say for example the one in Indonesia, perhaps they were only able to isolate, you know, the major pandamarylactam 3X and 3Y. Now, also another limitation, I have to tell you, another limitation in this isolation of Pandanus alkalis is that it is a little bit difficult to isolate them. The purification of these Pandanus alkaloids is quite challenging as well. So perhaps the one in Indonesia, based on my experience, the pandamarylactam 3Y is one of the easiest alkaloids to purify. Based on my experience, the pandamarylactone 1 is one of the easiest alkaloids to purify. Now, it is now difficult because, as you can see, the alkaloids, the Pandanus alkaloids, are actually diastereomeric mixtures, and it's quite difficult to isolate diastereomers. So it could also be one of the limitations why only in Indonesia there are only these alkaloids that were isolated. But in terms of the amount, you know, so I am referring to the Philippine samples. So in terms of the amount of the alkaloids, the pandamarylactonines would have the highest amount based on NMR studies. And based on the amount, say for example, I was able to isolate 150 milligrams, and that's a big amount. I was able to isolate 150 milligrams of pandamarylactonin A, B, and C, D mixture. So I could say that the Philippine samples, the pandamarylactonins, would be the major alkaloids. Yeah, in particular, pandamarylactonin A, that's a major alkaloid. So that is why the pandamarylactonin A is the focus of my research, because it is you know, in terms of the amount in the alkaloids, it has the biggest amount. Yeah.
Thank you. Is that answer your question?
I hope
Dea?
I answered the question.
Okay, maybe I get it, ma'am. Thank you for your answer, Mr. Thank you, ma'am.
Thank you. Yeah, it is very difficult to separate diastereomers, Professor Tan. So
Yes.
how did you do that? Which method do you use?
One of the methods that I use is that I use aminosilica gel chromatography.
Huh?
Okay, aminosilica
Mhm.
gel. And then I also, well, also MPLC. In MPLC, because in medium phase liquid chromatography, I can monitor the peaks, you know. So one of the techniques that I did is to collect it portion by portion. So eventually, because the peaks will overlap, right?
Yes,
So
correct.
what I did, my technique is that I try to collect the peaks, you know, portion by portion. I don't collect one peak all at the same time. because it is possible that I might be getting a mixture out of that. So say, for example, if I see that the peaks now starting to get out, or I try to monitor the UV, okay, because the UV absorption, if I see that the UV is now increasing, so I now try to collect, and then I change again the plus, etc. And then when I do, what is also good when I was doing this one is that I can easily monitor the purity via NMR. Yeah. So I think, you know, NMR is also very essential when you're trying to do natural products for research, because you can monitor right away if what you are isolating is pure or not. Yeah. So I think that's the technique that I use, you know. So I try to collect a little by little. If I am doing column chromatography, I also collect small amounts in the test tubes, and then I try to TLC. Yeah, because eventually these peaks will, you know, we will overlap. And
It will overlap, yes.
if you try to collect a big amount in a test tube, tendency is that you might be getting mixtures. Yeah.
Exactly. So it was really determination work, Professor
Exactly.
Tan.
Yes, exactly. Yes. Technique that I've learned based on my, you know, years of experience doing natural products work. Yeah.
And instinct as well.
Yes, yes, yeah.
All right. Ahmad is already here. Ahmad, it's your turn. What is your question? Yes, you can directly give question to Professor Tan, because it is in English, so it's better if you talk by yourself.
Bacakan
So
pertanyaannya.
go ahead, Ahmad. Yes, yeah.
Okay, thank you. Permission to ask initially why were you interested in the analysis of the Pandanus? What difficult were encountered when making the determinant of alkaloid compound for the Pandanus itself? Thank you.
Uh, I apologize. I think I didn't get your question. Once more, please. Sorry. Yeah. Can you repeat the question one more time, please, if you don't mind?
Bisa diulang, Ahmad, dengan perlahan-lahan dan jelas gitu ya. Silakan, Ahmad.
Jangan berlipat-lipat.
You ask, you really need me to read your question? All right, this is a question from Ahmad. Why were you interested in the analysis of Pandanus plant, and what difficulties were encountered when making the determination of the alkaloid compounds from the Pandanus itself? This is the question from Ahmad.
Okay, yeah. So first, why? Well, the Pandanus plants is one of my plants of interest. So I am interested in the Rubiaceae and Pandanus. Rubiaceae because I have a collaborator here in the Philippines. So I have a collaborator who is a Rubiaceae specialist. And in particular, I am interested in the Rubiaceae species because of the diversity of compounds isolated in this Rubiaceae species. So you have here a lot. It is quite interesting that in Psychotria, wherein Psychotria is known to have alkaloids, in the Philippines, we work on some Psychotria which would have an alkaloid, which does not have an alkaloid. Now, I am also interested in the Pandanus species because this is the plant which I first work on during my undergraduate. Okay, so in my undergrad, in my BS chemistry thesis, I work on Pandanus. In my master's thesis, I work on Pandanus. And then in my PhD, I also work on Pandanus. So I don't want to give up the Pandanus plants. And also because our university, through Dr. Maribel Nonato, is known to be working on this Pandanus species. So if you go to the Philippines and then you ask these top universities about Pandanus research, Pandanus research is synonymous to the University of Santo Tomas, where I am working right now. So I don't want to give it up because we want to continue. We want to continue as much as possible the legacy of working on this Pandanus species. Now, there's no special reason why I wanted to work on the alkaloids. Actually, I wanted to work on any type of compounds as long as it is biologically active. And currently, my research of interest is on these biologically active molecules which might have an effect against Alzheimer's disease. So that's my main concentration right now. It doesn't matter whether it is alkaloid, whether it's a terpene, whether it's a steroid, a coumarin, a flavonoid, etc. It doesn't matter as long as it has an activity. Also, it doesn't matter whether it is a known compound or a new compound. It doesn't matter, again, as long as it has an activity. Okay? And then what is your other question? What is the difficulty? Is that it? Sorry. Yeah. Yeah. So I mentioned a while ago that yes, there are a lot of challenges doing alkaloids research in particular in Pandanus because most of these Pandanus alkaloids are quite challenging to purify. So yeah. So I hope I was able to answer your question well, Mr. Amat. Really, I
Yes,
think. Thank you. Yes, thank you. Really, I think you are muted.
Okay. Do you hear me now?
Yes, yes, yes.
We have heavy raining now. So I think this is our last question from Dr. Dini Hadiarti. She is a lecturer from Universitas Muhammadiyah Pontianak, our neighboring university. Dr. Hadiarti, would you like to ask question directly to Professor Tan, or do you want me to read your question? Okay then. Yeah. So the question is it possible to control alkaloids such as their quality and quantity from raw materials of plant? The second question, as bioactive compounds for candidate of drug, how to make alkaloids suitable released in our body? I mean, do we need to derivate alkaloid or add other ingredients or something? Is the question clear, Professor Tan?
I think I have to answer one by one. Okay, so the
Okay,
first
yes.
one is that can we control the amount of the, well, not alkaloids in general, but in not alkaloids per se, but compounds in general. Now, well, if you try to collect a plant species in the wild, in the mountains or whatever, of course you cannot control the amount of those natural products. However, my experience in when I was doing my PhD is that I have a friend who is doing biogenetic engineering. So his work is on I forgot the name of the plant, but he is trying to culture, he's trying to culture plants.
Culture.
So because his main objective there is he tried to control based on he's trying to control the amount of a certain group of molecules. So I think that is possible via molecular biology process or biogenetic engineering. So based on what I saw in what he's doing, he's trying to control the amount of light. He's trying to control the amount of water that he puts in the plant, etc. In order for him to control the amount of these particular compounds. Yeah. But if it is in the wild, I don't think that is possible. Also, when you are trying to isolate, if you collect the plant from the wild, well, eventually the amount that you try to isolate would depend on your technique. Yeah. Okay. And then the second question, Dr. Rini, I forgot. Yeah,
As bioactive compounds for candidate of drug, how to make the alkaloids suitable released in our body?
Okay.
Do we need to derivate alkaloid or add other ingredients or something?
Okay. So in my work in neuroprotective studies, one of the considerations, that we have is that no matter how bioactive the compound is, since I'm doing work against Alzheimer's disease, no matter how bioactive, how good the activity, using the cells, using the whatever assays, if they do not pass the BBB, the blood-brain barrier, it wouldn't be an effective drug against Alzheimer's disease. So in my line of work, that is the one thing that we have to consider. The compound should have a good activity or a good effect, or it should pass, it should have an excellent blood-brain barrier activity. And how do we do that? That is where now, so if you try to see, or if you try to observe that a particular compound or particular alkaloid would have a good well, I'm speaking about Alzheimer's disease. This is the technique that we are actually using, no? So if we try to observe that a particular alkaloid would have a good neuroprotective effect, but it doesn't pass the BBB, we try to manipulate the structure. So we might change one particular functional group. We might add a sulfur containing functional group, etc., in order for it to have a very good BBB, blood-brain barrier activity. Also, based on the literature that I am trying to read, is that there are now some efforts in terms of the delivery system. So in terms of this, it can be converted, so these compounds can be synthesized using these various nanomaterials, which are good drug delivery system, or the DDS. The drug delivery system. It could also be one technique in order to enhance the biological activity. Because by itself, these alkaloids that we are trying to add, the Pandanus alkaloids, as you can see, are not very polar, no? So they're not that quite polar. So it depends on the disease that you are trying to target. So you also have to consider, the disease. Do you have to consider that the drug should be applied intravenous injection? In that case, it should have a very good solubility in water, something like that. So it depends on the biological activity. Of course, the ECS, in terms of the drug development, the ECS is that to develop like an ointment, because in ointment you just have to put it in your skin.
Yeah.
Yes. Yeah. Okay.
That's right. Thank you so much for the answer. I think that's the last question, and we are now moved to our final session. Thank you so much, Professor Tan, for delivering. I cannot say anything. I'm speechless. That was so plenty of information. I hope that all the participants here can receive it well and inspire them to be a better scientist, a better person as well.
I hope to have a collaboration with Joshua Rini in the future. I hope we can
I would
collaborate.
love to. I would love
Yes,
to.
yes.
This is not the end.
Are you still
I would
doing
love to.
Are you still doing the phytic fungi in because I know your work is in fungi in Germany, right?
Yes.
Yeah. So
Yeah.
you doing the phytic. So I hope we can have collaboration in the future.
Yep. Yep.
Yeah.
I will contact you again after this as
Yeah.
soon as possible.
Thank
Thank you so much, Professor Tan. So
you.
I'm now give to MC De Aninda for the next session.
Okay, that's the lecture from Mr. Tan. Thank you for your very good lecture. The next agenda is certificate and photo session. To the operator, please, and to Mr. Tulus, would you please hand over the certificate simultaneously?
Okay. Thank you very much, Professor Tan, for the knowledge shared, especially for organic chemistry. Hope it is useful for our students and all participants to appreciate these activities as guest lecturer. Let us give a certificate to Professor Tan with respect. Here the certificate, Professor Mario.
Thank you so much again. Thank you so much for the invitation. Thank you so much to the faculty and students of the University of Tanjung Pura, the Department of Chemistry Education. Thank you so much for this
Thank
opportunity
you.
to share my knowledge and to share what we have done in the chemistry of the Pandanus alkaloids. Thank you so much. Thank you.
Thank you. Once again, thank you very much for your time. Hopefully, next time can provide new knowledge for us. Thank you, Professor Tan.
Okay, thank you, Mr. Tulus. Next, maybe we can take a photo session. To the operator, please, for page 1. 1, 2, 3. Okay. Next, page 2. 1, 2, 3. Okay. The last page. 1, 2, 3. Okay. Thank you. Ladies and gentlemen, we finally come to the end of today's agenda. Let us close the lecture by reciting Hamdalah. Alhamdulillahirabbil alamin. Those are the agendas where we have present to you all this day. May all of us get the value and benefits from the lecture. Thank you so much for your attention. See you on the next occasion. Wassalamualaikum warahmatullahi wabarakatuh. Have a good day, everyone. Thank you.
Thank you, Professor Mario Tan.
Thank you, Ani.
Thank you, Prof.
We are looking forward to see you again.
Yes, yes.
Palam na.
Thank you. Bye-bye. Bye-bye.
Bye.
Bye.
Well.
So, terima kasih semua.